And maybe Aaron, you may want to share your screen.
Sure.
Yeah. Yeah. So his maybe most recent work, and the topic of his talk today is stochastic. Can I see your title? Yeah, processes as a scientific instrument, efficient inference based on stochastic dynamical systems. So please join me in welcoming Dr. Aaron King.
Thank you so much, Jay. It's real honor and a great pleasure, actually, to stay up a little bit late to have a chance to talk to such an august and distinguished group of people at such a fascinating institute. Jay was telling me a bit about all the very exciting things that are going on there, and I've really got my eye on what you guys are doing there in Korea. So I wanted to kind of talk to you about a few things. As Jay says, the techniques I'm going to talk about today in sort of broad terms are useful in a wide range of different systems, dynamic systems in biology primarily, but really beyond biology even. And so I'm particularly interested in your impressions, your questions, and your insights into the kinds of problems that these approaches may be useful for, the limitations of these approaches, and anything else you have to observe. So before I forget, I wanted to point out a number of the people that have contributed to the work. So there's a number of postdocs, Felicia Magpente, Mathieu Dominic Dessel, who's currently in Berlin, Carlos Breto, who's in Valencia in Spain, Jenny Levine is at Emory University, and Dai Hai He in Hong Kong. Mm-hmm. My long-term collaborators, Ed Ionides, Pedro Hani, and Mercedes Pascual, funding from various places and institutes that have helped make working groups possible to produce these results. So, you know, I'm going to talk a lot about models, right? And using models for particular purposes. But if we think about it broadly, of course, we're all aware that models have many purposes to which they're put. You know, in early days, we use them to just organize data, to keep track of different kinds of data, to develop theoretical principles and work out ideas that are hypothetical about the way that biological systems work. They play an extremely important role, as we're all very much aware now, in guiding policy. Whether or not they're based on sound principles, they find a role in guiding policy because we need guides to policy. But I want to focus today on really the true scientific purpose of models, which is to help us explain observed patterns. So I'm interested in models as they help us to understand biological systems, to understand the mechanisms that are responsible for patterns we see. And so really we can think about mathematical models from this point of view as a kind of scientific instrument, like a microscope or a telescope. They are tools for examining data. And if you think about what kind of tools they are, they're a little bit like a microscope or something, in that they focus our attention on the discrepancies between hypotheses and our observations, hypotheses and reality. They tell us which discrepancies, which deviations from our hypothetical explanations are large and which are small, which are surprising and which are to be expected. And thus they help us focus our attention on the places where our models can be improved. How do we go about quantifying discrepancies? I already hinted that the key metric involved in quantifying discrepancies is the metric of surprisingness, which is a probabilistic concept. And so we fall back on the key principle in statistics, which is modeling the error, modeling all of the deviations. So when we think about studying intact biological systems, ecological systems are a wonderful example, but epidemiological systems in real life, in a free behaving population, as another example, how do we go about making inference about the mechanisms, right? We can't perform experiments in any real sense while keeping those systems intact. And so dynamics are a particularly informative approach, right? If we can see how a system behaves dynamically, we can test hypotheses about various putative causal mechanisms. So in particular, we can formalize hypotheses about causal mechanisms as dynamical systems models. And then we're left with the statistical problem of inference. So how do we go about estimating the parameters, the unknown parameters that are invariably present in such models? How, when we have multiple competing hypotheses, do we go about deciding which of them is better supported by data? And when we have rare and precious data, as we typically do in biological systems, we never have enough data. How can we make sure that we're extracting the most information possible from the data that we have? And finally, when do we know when we need a better model versus better data? These are the kinds of questions that come up when we use mathematical models as scientific instruments. So again, the key principle in thinking about all of this is that we must probabilistically model the error, and that we can immediately break down the kinds of discrepancies that arise into two different categories. One of them is measurement error, which of course is in part due to actual error in measurements, right? Sampling error, biases in various kinds of measurements, just noise in measurements. But even if we have very good measurements with very little error, the very finite precision itself, the fact that our measurements are made with finite precision, it must be accommodated in terms of measurement error. Looking at biological systems themselves, there's always also process noise. So there's stochasticity present in the biological systems themselves. And those come from a variety of different sources, from fluctuations and trends in the environment, the various variables that describe the environment, heterogeneities in various populations of cells or organisms that are left out of the models. Other forms of model misspecification, which are not necessarily intrinsically stochastic, can be accommodated using stochastic models. Allow the models to fit the data even in the presence of misspecification. And finally, there are always many, many variables that we can't include in the model or don't want to include in the model. And only through stochastic formalism can we allow those unmodeled variables to sort of play. So here's what I want to talk about today. With that introduction, I'm going to first just introduce a simple class of Markov models for epidemiological processes. But as Jay was saying, we can extend these to lots of different kinds of systems. To give you an idea of the kinds of questions that we've been asking, just a flavor for the kinds of issues in the models, I'll give you a couple of examples just very briefly, and then talk about a class of models that allow us to address these questions, the so-called partially observed Markov processes. Then I'll give a quick overview of methods for inference that are available for these things and some practical considerations, and finally conclude with some attempt at synthesis. So the class of models I want to call your attention to is a familiar one to us all now. I think if you in Korea are like we are here, essentially everybody with an education is an amateur epidemiologist at the moment. And of course, right? And you know, these compartmental models are ubiquitous in epidemiological theory. Right. So, you know, this is the very simple caricature where we divide a population of hosts into different categories. In this case, those hosts that are susceptible to infection, those hosts that are actually infected and transmitting infection, and those that are recovered and perhaps immune for a bit. And if you know, in addition to those basic processes of infection, let's see, can you see my cursor?
Yes, we
So in
can.
addition to the process of infection, whereby susceptible individuals become infectious, and the process of recovery, where infectious individuals recover, we have the processes of death and birth, of course, which add individuals to these compartments or remove them. And this can be translated, if we sort of think about the numbers of individuals that are inside of each of these compartments, and we focus on those, we can abstract away and just look at the dynamics of those compartments. If we label them, we keep track of X and Y, the occupancies or the numbers of individuals in each of these compartments and the rates at which individuals move amongst them, then that allows us to write down, for example, well, we can write down various kinds of formulations of the models. A familiar one is a stochastic differential equation formulation, where we keep track of the numbers of individuals moving along each of these arrows, either from X to Y, with n of x, y, and write that down as a sum of a deterministic component and a stochastic component here, with dw playing the role of for example, a normal or a gamma driving process. Here, this is nx0 is the death process. Well, I guess actually it should be y. Here, describing the process of death from this category. Anyway, so this is one particular example of an instantiation, a particular form that this model might take as a set of differential equations, stochastic differential equations. And then with those, of course, we can reconstruct the dynamics of the state variables X and Y. So we end up with a Markovian process. Now, this can be made arbitrarily complex. This is a very, very simple model with just a couple of variables that can be made arbitrarily complex. And we'll see some of that as we look at some real examples. So the first example I want to talk about is from a study that I've been involved in for many years now, looking at cholera in its endemic home. So all but one of the pandemics of cholera that swept the globe in the last 2 centuries originated here in the sort of low-lying regions just north of the Bay of Bengal, and in particular in Bengal. And we have a collaborator who digitized long time series showing cholera deaths during the period of British rule in this region. So here's just an example of what these look like. These are monthly death counts over a 50-year period, and in 3 different districts in Bengal. So the dynamics are quite complex. They show annual or seasonal, strongly seasonal dynamics with sharp peaks in some regions every year. In fact, in many regions, twice per year, which is an interesting pattern in and of itself. They show multi-amplitude or multi-annual oscillations, or what look like them. Quite a lot of random variability or sort of harder to get a grip on variability here. And as we move further, so this is in Dhaka, one of the larger districts. Going north, we see regions where there are extended periods of fade out or very few deaths, punctuated by extremely large outbreaks. And closer to the seashore, we see long periods of time when there are years and decades go by where there's at least some deaths every month. So the dynamics are quite complex. The questions that are of interest here are to do with the relative importance of the various routes of transmission. One major set of questions, right? So we know that cholera has an environmental reservoir. The bacterium is found in association with various copepods in brackish waters. But we also know that transmission can occur very rapidly between individuals. How important are these 2 different pathways in the ecology of cholera in this region? the duration of infection-induced immunity is another big puzzle, right? The vaccines for cholera are notoriously short-lived in their efficacy. What about natural infection, and how long does the immunity last to natural infection?
Mm-hmm.
Then there are the patterns that we see in the data in terms of both the seasonality within the year. How is it that we get this complex seasonality of multiple peaks per year? And what is responsible for the multi-ennial cycles, the multi-year cycles that are there? There's been putative roles suggested for various decadal scale climate drivers. And then there's also interesting questions to do with the wild ecology of cholera and the interactions between cholera and the phage in the environment.
Mm-hmm.
So this is just an example of a variety of different scientific questions. And so one way to approach it is to try and tailor mathematical models that can capture various hypothetical answers to these questions. So starting with something quite similar to the compartmental models I drew before, we need to add some pathways to account for the fact that once recovered, an individual can become susceptible again. The immunity is not permanent for cholera. There's human-to-human transmission, but there may also be transmission from an environmental reservoir. So both of these things can contribute to the force of infection.
Mm-hmm.
In addition, there's seasonal modulation potentially in both of these things. Both of these things can be functions of time. There are deaths that are caused by the infection, obviously. About 50% of the severe cholera cases resulted in death during this period. It's thankfully much lower nowadays. But the data we have we actually observe none of these variables at all, right? The only data we observe are the deaths. And finally, as I was saying, there's the potential for infectious individuals to shed bacteria into the environmental reservoir where it can interact with bacteriophages. So if we look at it from a mathematical point of view, the transmission models that result from trying to capture these mechanisms are nonlinear, obviously. They're stochastic. They're highly non-stationary due to the presence of various environmental drivers. And these extrinsic drivers are themselves environmental variables. So the models that I've sort of very crudely described to you are attempts to capture the mechanisms, to describe these different mechanisms. And so the questions that arise are, how well do the various models that lie nested inside of those diagrams that I described to you, how well do they explain the data? And what kinds of insights can they give us into the nature of the system's dynamic self-regulation? Mm-hmm. The key thing I want to call your attention to here is that the structure of the models that we've formulated here correspond very closely to the questions of scientific interest, right, and not to any particular category of tractable mathematical model. So if we are following our scientific interest, we very quickly arrive at mathematical models that are extremely difficult to treat analytically. Mm-hmm. And so, as I would argue, as scientists, we need to be able to, we need techniques for dealing with models that are inconvenient from a mathematical point of view. Mm-hmm. All right, I think I've said this already. Mm-hmm. Let me give you a second example. This concerns the ongoing resurgence of pertussis. So pertussis is another bacterial infection. It's a respiratory infection. It's one of the major causes of vaccine-preventable death, especially in children worldwide. And despite the presence of highly effective vaccines and even very high coverage in many countries of the world, pertussis is making a comeback. And so this is what that looks like. This is just data from the American state of Massachusetts, where we see these are annual cases on a log scale. You can see that when the vaccine was introduced in the 40s in the United States, mm-hmm, there was a rapid drop in the overall incidence, but since the late 70s, it's been coming steadily back. That's also manifest in a shift in the age distribution. So in the pre-vaccine era, what we see are a lot of cases in children, preschool and in early school age children. But in the more recent period, there's still a mode here with the infants, too young to be vaccinated. But we see now the majority of cases are in teenagers in these highly vaccinated regions. But the picture is quite complicated. You might think, some various explanations might occur to you to explain these patterns. For example, failure of the vaccine or waning of the vaccine. But if you look across the world, the pattern is far from clear. So this is a rather complicated figure. Similarly, on the log scale, I'm showing yearly incidence, just as I showed before, over a shorter period of time from 1980 to 2012. But we have a bunch of different countries. This is all WHO data. And the black lines show the incidence, the annual incidence on a log scale of pertussis in these different countries. We've done just some very, very crude descriptive statistical analysis to determine whether we see a decreasing trend shown in the blue lines, like we see here in the early part of the Spanish data, or the Danish data, or the late part of the Italian data, or all the way across in China, or increasing like we see in most of Australia, most of the United States, and so on. And then there's a vertical dashed line which describes the switch in vaccine from a whole cell killed or inactivated whole cell vaccine to the more modern vaccine, which is an acellular vaccine containing just a few proteins. And you can see that in the United States, for example, in the United Kingdom, where the switch to the new vaccine essentially coincides with a change in the trend in pertussis, and the same in Spain, but in Sweden, for example, the trend is exactly the opposite. It was trending upward and then reversed that direction. The same in Canada. And in places like the United States, it's just been increasing on both sides, and similarly in China on both sides. So it's a far from clear pattern of what's going on and whether the vaccine has anything to do with it. So the scientific questions here are to do with the vaccines, whether there are changes in the vaccine efficacy associated with the shift from the whole cell to the acellular pertussis vaccine, whether the pathogen is itself evolving in response to the vaccine to evade antigenic evolution, to evade protection, or some other characteristics of the bacterium changing. Whether there's some other various explanations to do with asymptomatic transmission or loss of natural immune boosting, or even merely increased sensitivity of surveillance. In all of these explanations, the key unknowns that we are forced to try and estimate are things like the durability, the duration of effective vaccine-induced immunity, and the relative efficacy of natural immunity to vaccine-derived immunity. So one can try to get at these questions by adapting a model or devising a model that incorporates the various mechanisms. So here's a model of vaccine failure, right? So individuals that are born can be vaccinated. So some fraction of the individuals that are born are vaccinated, they enter this V class. Those that are unvaccinated, or for whom there's primary vaccine failure. They're vaccinated, but there's no protection that ensues, enter this susceptible state. And if they're in the susceptible state, they can become infected by following this path. First, a latent infection, then a transmissible infection. They can recover. But if they're vaccinated, there's two ways the vaccine can fail. The vaccine can sort of wear off through time, can wane in its efficacy, and then they become susceptible and can become infected and move down this progression. Or the vaccine may may be imperfect in the sense of a leaky vaccine. It may afford a partial protection only, so that there's some reduced chance of becoming infected, but that some individuals who are vaccinated do become infected. So there are three different vaccine failure modes here. One is a primary failure, failure in take. One is leakiness, that is failure in degree of protection. And one is waning, which is failure in the duration of protection. And of course, the reality can be some combination of all of those three. Importantly, individuals that are previously vaccinated but become infected might differ in their transmissibility, right? So when we think about the overall force of infection, the rate at which individuals become infected, it's clearly a function of individuals that are infected here, but that it can also, there's a parameter theta here to describe the relative transmissibility of these sorts of infections.
So theta is less than 1, typically, I guess?
Theta, well, you could constrain it to be less than 1. It's conceivable it's bigger than 1. Probably it's less than 1. But it might be equal to 1, for example. It might even be 0, right? Anyway, so, but that's an unknown parameter. And moreover, the reports, right? So the data come in the form of case reports. Some of those are in post-vaccinated individuals, and some of them are in naive individuals. But it's not clear how observable the post-vaccination infections are. So we assume that post-vaccination infections are observed at some reduced rate, where eta is the reduction in the rate of reporting. So with such models, we're interested in, can we estimate the nature, that is to say, what kind of failure occurs? Is it primary vaccine failure? Is it waning? Is it leakiness? And can we investigate the durability, or can we estimate the durability of vaccine-induced protection? So there's a bunch of models nested inside of this. Which of them explain the data well? And so to deal with that, we face questions of parameter estimation and model selection. The data I want to describe to you to begin with are drawn from the US state of Massachusetts. And just to kind of give you some to look at, these are what the data kind of look like. So these are monthly case reports, and they're stratified by age. So up here in light blue, we have the infants less than 1 year. Then we have 1 to 5 year olds, 5 to 10 year olds, on up to 40 year olds plus. You can see what the dynamics look like. There's a seasonality to the disease. And there's also multi-ennial cycles, quite common in immunizing infections. If we just look at the age structure, just normalized by the total number of cases, and just see what fraction are in each age group, we can see that early on, so this is 1990 here to 2005, in the early period, there's a substantial number of cases among the infants, and that number decreases through time, whereas the number of cases in the teenage population here, 15 to 20, actually increases, along with infections in older individuals. And you can see that in a variety of different ways. Okay, so in order to deal with those kinds of data, we have age information in the data. So that complicates the model even more, right? Because now, on top of all of this infection status structure that we have here, with all of these different boxes, we have age structure as well. So we can visualize that by just having a stack of these boxes, one for each age group, each age category. And so we have to think not just about vaccine leakiness from vaccinated individuals, but that's potentially different with age. And the waning, of course, interacts with the age of these individuals. The complexity is heightened by the fact that we have data as covariates. So we have time-varying covariates. So vaccine uptake, we have data on that. We have changes in population size and changes in birth rates as well, making actually pretty substantial changes over this, even this just 15-year period, drops in birth rates. So all of those things are changing in time. In addition, the human population, of course, is not a panmictic homogeneous thing. In fact, different individuals of different ages mix with one another in characteristic patterns in the United States and in most developed countries. And in countries where education for children is mandatory, you often see a pattern that's like this. So this is a kind of a cartoon, but what we have here are this is an age structured contact matrix. We have age of contactor along here, age of contactee along here. And then these contours represent the magnitudes of the contact. So what you find in developed countries is to a first approximation, and if you're a school-age child, almost all of your contacts on a daily basis are with individuals that are almost the same age as you, right? Your classmates. And so that's what we see here. It's a little funny what's going on here. It looks like this guy is reading this guy's paper. That's a bit of a problem. And then what this man is going to do with this child, it's a bit scary. It looks like he's about to beat that child. But anyway, so off the diagonals, we have the intergenerational contacts.
Let's see.
And then contact rates are much smaller in the older age groups. These are more conventional representations of the data. When we have data from two different actual data sources and different methods of reconstructing the contact network, and they agree in broad strokes and differ in some of the details. Right, so you can see the sort of strong diagonal here, and then the off-diagonal components in this data from Europe. The data that's reconstructed on the basis of the Massachusetts census is a little bit less resolved, especially in the older age groups, but you do see this very strong assortative mixing here in the younger age groups as well. So we can put these data into this model in order to try to address these questions. So again, we end up with a model that is highly inconvenient from a mathematical point of view. None of the, very few of the densities that, probability densities that you would want to evaluate here in order to perform inference on this model are available in any kind of computationally convenient form. What you can do, however, is simulate from these models. And so I want to talk about a class of approaches that involve, that are based upon simulation. So, but before we do that, I want to just point out that all of these models that I've been talking about so far all fall within a class of so-called partially observed Markov processes. So just in a nutshell, this is what we have. We have some true but latent nonlinear stochastic process that is nonlinear. I've sort of diagrammed that by having a circle of causality here. This is a system that evolves according to its own rules. It's affected by environmental forcing, and in order to observe it, we have to, the states of this Markov process here are filtered through some kind of a measurement process, and that's how the data are generated. These blue arrows show the sort of direction of causality in this kind of a structure. The information flows in the opposite direction. And so if we want to get information from the data about the thing that we're interested in, we can only do that via the measurement process. So formally speaking, these partially observed Markov processes are defined in terms of a set of observation times, a set of data. This can be multivariate data. Some unobserved Markov state process, this is a latent state process, some measurement process, and there are therefore some probability densities that are involved. We'll let little f here refer to the probability density of the transitions of the unobserved state process, and g refer to the density of the conditional density of the measurements given the states. And of course, there has to be some sort of initial density as well. So the basic problem in doing inference on this kind of a problem, this is also called a hidden Markov model or a state space model, although it's not a linear or a Gaussian state space model. In general, it's a nonlinear, non-Gaussian state space model. So the central problem, of course, is the following, right? If we want to, well, here's the full joint probability density of the x and y's together. and it's just a product of these different densities. The Markov property allows us to factorize it this way. And if we want to perform inference, the central object is the likelihood function, right? Which is just the density, the marginal density of the y variable, the observed variable, evaluated at the data, viewed as a function of the parameters. In order to get that, we have to essentially integrate out across all of the unobserved latent variables. And so this is typically a very high dimensional integral and extremely difficult to evaluate, and so that's the central difficulty. So everybody who wants to work with this has to come to grips with this in some form or another. Now, so let's talk about these mechanistic models here. If we want to compute the likelihood, if we want to maximize the likelihood or perform Bayesian inference based on this likelihood, we essentially need to integrate over this huge space of x's. And so the traditional solution to this thorny mathematical problem is sort of to be clever, right? So one can either simplify the model to make it statistically tractable, right? Which means that you throw out you make it more difficult to address the scientific questions that you want to address. Or you could devise and tune some sort of inference algorithm, for example, the classical choice is an MCMC sampler, and you could tune it specifically to that model so as to make it efficient at sampling from that, from those distributions. But when we do this, if we have multiple models in mind, we have to ask ourselves, are we testing our hypothesis or our scientific question, or are we possibly also testing a statistical method, right? If we put different amounts of effort into tuning different models, maybe because we like one model more, or we're more familiar with that one model, another model is less well understood, then we run the risk of having that color our inference. Stepping back one step, it's even perilous, I claim, to invest too much time in any one biological model. There's the Pygmalion problem, right? The Pygmalion was the sculptor who fell in love with his own sculpture. And if we invest a lot of time in our models, we have a tendency to fall in love with them. And as scientists, we need, of course, to resist this temptation.
Right.
The traditional antidote to this has been the method of multiple working hypotheses, right? To have multiple models, so then you're not attached to any of these. But in order to be able to work with multiple models, we need methods that don't depend upon the precise form of the models. Mm-hmm. This leads to something that we call the plug and play property. An algorithm, a statistical algorithm, has the plug and play property if it does not require one to ever evaluate the process model density. And this is typically the hardest thing to do for any of these models, to actually evaluate the latent state transition density. Plug and play methods, instead of requiring you to evaluate that density, they access the process model only via simulation. And this, because simulation is typically quite easy, no matter how complicated the model is, this puts minimal restrictions on the form of the models that we can study. So let's talk a little bit about some of the methods that are available to us. So I can divide those methods into two basic categories. There are methods that are based upon features of the data, and there are full information methods that are based upon the full likelihood. Feature-based methods include a number of methods that you are probably have heard about or even worked with, including approximate Bayesian computation. There's some nonlinear forecasting methods that are and attractor reconstruction-based methods. Probe matching and synthetic likelihood are also have been developed to do this. And all of these methods are alike in that they reduce the data to some set of summary statistics, and then through simulation, if you can compute a set of summary statistics on the real data, you can also compute them on the simulated data, and you can compare the model to the data that way. Full information methods tackle the problem of computing the likelihood itself. And basically at this point in time, there's essentially one efficient algorithm or one class of efficient algorithms for accessing the likelihood in this context. And this is the so-called sequential Monte Carlo, or the particle filter. I'll talk about it in a little bit. Based upon this, one can do inference either in a Bayesian mode through something called particle Markov chain Monte Carlo, or through iterated filtering. All of these methods are available in a software package that I maintain myself, and is available, freely available to everyone. So just to give you a flavor of some of these different things, synthetic likelihood, here's how it works, the very simplest form of it. So given some parameters and some data, we collect a set of summary statistics. These are functions on the data space into d-dimensional Euclidean space, and we compute these observed probes by evaluating those functions on the data. Then we simulate a large number J of data sets and compute the simulated probes on each of those data sets. And then we can compute a synthetic likelihood. Here, this is based upon a normal assumption, assuming that the distribution of these statistics is normally distributed, but it can be, you can vary that assumption quite easily. And through simulation, you can compute this synthetic likelihood. And the objective then would be to maximize the likelihood over the values of theta, maximize the synthetic likelihood, or perform Bayesian inference using the synthetic likelihood as the driver there. The full information methods, as I say, are all based upon a particle filter. And the particle filter is essentially a sequential algorithm. It begins by sampling a large number of particles, a large number J of particles from the initial latent state density. And then it's a sequential process of predicting using the latent state process and evaluating those predictions in terms of the likelihood of the data under those predictions. And there's an implementation of Bayes' theorem here in the form of a weighted resample. And this gives us, importantly, an estimate of the likelihood. It's a Monte Carlo estimate, but it's an unbiased estimate of the likelihood. One can use that essentially in, here's a Metropolis Hastings algorithm using that likelihood computed by means of the particle filter. That's called particle Markov chain Monte Carlo, and it's the most efficient algorithm currently for doing Bayesian inference in this context. If you want to maximize the likelihood, the best way to do it is to use what we call iterated filtering, which essentially involves iteratively applying the particle filter in a manner that with some supplemental noise that is annealed. So there's some analogies to simulated annealing here, but I won't go into the details. Putting these methods into practice, we have these various methods that are available and have been, our group and many other groups have been using them. I want to say a few words about how we use them in practice. So there are various criteria for comparing models, right? And so when I talk to students about this, these are some of the model comparison criteria that I run across a lot. So, and actually not just talking to students, also in the literature, we see this. So the best model is the one that agrees with the already published results. And this one, it's funny when you see it written down like that, but this is something we quite commonly see in the literature, is that the conclusions of a study are compared against conclusions drawn in previous studies. And to the extent they agree, it's considered evidence of success. But the question I'd like to ask is, what are the prospects for actually making revolutionary discoveries, revolutionary scientific discoveries, if this is a criterion? Obviously, the best model is the one that I can estimate before my thesis is due. And this is a joke, but I mean, there are real issues to do with the amount of effort that is put into these problems. Solving these kinds of problems it involves a non-trivial amount of computational effort, and that can be difficult to square with other kinds of of time pressures. The best model is the one that best agrees with my own preconceptions. I think that one is often present in many analyses. The best model is the one that makes my supervisor the happiest. Here's an interesting one. An identifiable model is better than a non-identifiable model. If I can estimate all the parameters of my model, that makes it a better model. And this is actually incorrect. That there are problems if your goal in your estimation exercise is to identify parameters. Then if you can't identify it clearly, you will have failed. But that doesn't necessarily say anything about the ability of the model to explain the data.
Right.
And finally, we often see this kind of confusion that the best model is the one with the narrowest confidence intervals. Anyway, these are kinds of common misconceptions. So on the other hand, there are various model comparison criteria that do make sense. So various ways of looking at how well the model fits the data. And some approaches that we found particularly useful are to compare the actual likelihoods. People often ignore the likelihoods, the value of the likelihoods. They only look at relative values of likelihoods. But the absolute values are useful, and they can in particular be compared across widely disparate models. So often to benchmark models, very simple models, even non-mechanistic models that provide benchmark likelihoods. And then there's the question of, does the model fit well? Acknowledging that the best fitting model is not necessarily a model that fits the data well.
Right.
Much more important, I think, than just quantifying how well the model fits the data is how does the model fit the data? So which aspects of the data are explained and which aspects of the data are ascribed to noise? So this goes back to the observation I made at the very beginning, that the function of the model is to identify and quantify the relative importance of the various discrepancies. And so that's what we're looking at here. And finally, there are various ways of getting at whether the model is adequate, in the sense that it's capable of actually explaining the data, right? Whether the data are actually consistent with the model assumptions, and whether, if the model were correct, whether the data would be a plausible realization of the model. One can go beyond this, and we're starting to go beyond this in the number of different infectious diseases, not having much success, as I would say, as a community, in actually making accurate out-of-sample predictions, but that's a much more sound way of trying to really evaluate how the models work, how well the models work. Often it's not acknowledged that the parameter estimates themselves, right, can be viewed as predictions about from the model that can be compared against independent data if those data exist. Another important criterion to do with models is the fertility of the model. Does the model make testable predictions? Does the model lead to predictions that can be tested through other data gathering activities? So just to quickly kind of wrap up a little bit, to go back to our cholera model, we were, this is one output of the analysis we did using that family of models. This is a profile likelihood plot. So for each value of a parameter, in this case, the duration of immunity, the mean duration of immunity, we maximize the likelihood over all the other parameters, and that's what we're seeing here. And so we see that there's a certain range of this parameter, which is compatible with the data, and then anything larger than that is incompatible. And in fact, so this is a very strong prediction of the analysis, and it says that if that model is going to have any chance of explaining the data, we've got to assume that the duration of immunity is very, very short. And there's all kinds of reasons why that's a problem, right? That if you go back to the medical journals at that time, it's very rare to find anybody who survived cholera and had another attack of cholera at all in their life, and certainly not, very few of them within a very short period of time. But this, we're talking about an order of weeks. you can get some insight into why that is by looking at these profile trace plots. So here we are varying that parameter across this range. Here's the likelihood again, the log likelihood. And here we're seeing another parameter and how it changes to compensate for the change in this parameter, right? So the change, as we change the immune period, the case fatality, for example, goes from being high when you have long-lasting immunity to very, very low if the immune period is short, as the data seem to want it to be. And so that suggests that what's going on is that the model is only able to, it wants to have very short-lived immunity, but it's coupling that with very, very low fatality. So if we couple that with the fact that, as I said, severe cases of cholera in this period had a fatality of about 50%, this is saying that most of the infections, the overwhelming majority of infections, are actually unobserved infections. So presumably asymptomatic or in some other way unobserved infections. And going back to the dynamics, you can see why it is that the model needs to fit the data this way. If we had infections, epidemics that are this explosive, they would very rapidly deplete the number of susceptible individuals in the population, right? If immunity was long-lived. And so you would not be able to see these kinds of recurrent epidemics every single year if herd immunity would be built up long before that. So the only way you're able to continue to get these outbreaks is if a small portion of the population is made immune. And but then you have to explain how the epidemic comes to a stop at all. And so the model does that through an assumption of immunity. So you can go back and say, well, this model assumes that every infected individual is alike. Essentially, the Markov property tells us that every infectious individual here is the same as every other one. Is it necessary that all the infected individuals be equally infectious? The plug and play property allows us to entertain a model to just change the model, introduce a new effect into the model. Now we can have some individuals that are totally asymptomatic and lose their immunity and become susceptible again, as opposed to the more severe ones that are at risk of death that develop some immunity. And when you do this, you can reconcile the duration of immunity in severe infections and the case fatality information with the parameter estimates from this. So there's a sort of following your nose along from one hypothesis leading to another that is facilitated by this plug and play property. When we look, I guess I'm going to brush over this, but here's a similar plot from the pertussis study. What we see here is actually a wide range of a parameter. In this case, this is the relative reporting rate parameter, where the likelihoods are quite flat. The log likelihoods are quite flat. So this is telling us that the data don't have an opinion on this parameter inside of this range. And from that, when we look at this other parameter here, we see it's even more pronounced, extremely flat. Extremely flat likelihoods. And this tells us, of course, the data don't have an opinion on the value of that parameter, but it's extremely useful to know for sure when you can't know the answer to your question. So here we've asked a question of the data, and the data have very clearly told us that they don't have an answer. That is extremely useful. So I won't say more about the pertussis case because I'm out of time. At any rate, it was just an example. I'd say, to summarize, that having worked with various of these methods over quite a number of years, that they all have their merits and their problems. So synthetic likelihood throws away information because it reduces the data to a relatively small collection of summary statistics, and yet in so doing, it gains robustness. So that can be a big advantage. It's difficult to know how much information it throws away. And in order to actually assess that, you have to compare with a full likelihood approach. And that So you never really get away from looking at the full information. But in addition, a problem with this approach and with ABC and other feature-based methods is that you have a wide range of choices you can make for the different summary statistics or probes, and they are consequential, so they can affect the inference. So that can be a problem.
Mm-hmm.
Iterated filtering, which is a likelihood maximization technique, converges much, much faster than particle Markov chain Monte Carlo. And so there are, of course, issues here between a frequentist and a Bayesian inference style, and there are various reasons for choosing one over another. But often one doesn't have to choose between those, make any absolute choice, and can make use of the strengths of each in the service of the other. So in particular, for example, to do Bayesian inference, you'll need to use particle MCMC, but it's probably a good idea to initialize the chains, the Markov chain Monte Carlo chains, using iterated filtering. These probes, do turn out to have a great value for post hoc checks for model adequacy. So I can talk more about that if there's interest. So what I think is the best way forward is an integrated approach using iterated filtering and profile likelihood to identify the heights in the likelihood surface, using particle MCMC to obtain Bayesian posteriors and smooth state trajectories if those are needed. And independent parameter estimates can be used to diagnose misspecification, and probes as an additional way of looking into the predictions of the model.
I see.
So just to conclude, the essential element in all of this is thinking probabilistically about the error, both the process noise and the measurement error, that the scientific hypotheses we have in real problems lead to mathematical models that are not very convenient, but plug and play inference methods facilitate effective investigation of those hypotheses. There are efficient methods available, but there is a continuing need for methodological development to improve computational efficiency and to accommodate new kinds of data, for example, genomic data and so on. Okay, so that's all I have, and I'm afraid I've used up every last minute of my time. But if any of you do have time to stick around and ask questions, I'd love to hear them, and I'd love to get your impressions. Thanks very much for your attention.
Thanks, Aaron. Yeah, we do have time indeed. Thanks for the great talk. Audiences, if you have any question, please let me know. Please unmute yourself, and you can ask the question directly to Professor King. So while waiting your question, there was one question from chatting room. That's the case of SARS-CoV-1. It was more lethal than SARS-CoV-2, and that's why it disappeared. I don't know. It was the question for one part of your talk. So
right. Yeah, it's more of a comment actually.
I see. Okay. Any question from audiences?
Hi, Professor King.
Hi.
Could you explain more about the plug and play inference?
Yeah.
Yeah, go ahead.
Well, can you be more specific?
So yeah, how can we, for instance, maybe, I guess with your GitHub, we can do that kind of thing, right? So maybe a little bit tutorial how we can do it if we really want to do that.
Right. So, so the basic idea. So is the basic idea of plug and play inference you want me to spend a bit of time on, or do you want to talk about? Okay. Yeah. So the basic idea is that, these mathematical models are essentially probability distributions, right? And I wrote down the joint probability distribution. And with the probability distribution, there's sort of two mathematical operations that you can perform. Well, there's more than two, but there's two big ones, right? One is you can evaluate the probability density, right? And the second is you can draw a random sample from that probability distribution.
So probability distribution of the models or parameter models, I guess. Oh,
Of all of these interesting models,
oh,
evaluating the density is very difficult,
right.
but simulating is relatively straightforward.
Mhm.
So a method, an inference method that you can use that only needs simulation is going to be able to accommodate a much wider class of models than one that requires you to evaluate the density. So for example, Markov chain Monte Carlo in its traditional implementation, or its most common historical implementation, requires one to evaluate. It's all about evaluating densities, right? And that can be extremely difficult to do in this context. But simulation is quite easy to do in this context. It's typically the first thing that you can do, right? It's the easiest thing to do. So a method, right? So that's what plug and play methods are about, and that's what they exist. And there are, there are more. I mean, I listed the main methods there in terms of the way that they, in terms of the main inference approaches. And there are multiple implementations of these in various places. I gave a link to the POMP package, which is a package for an R package for partially observed Markov processes. And all of the methods that I highlighted there are implemented in that package. So that's one place you can go and find these methods implemented. So that package gives you the ability to specify a model by giving various computational procedures for simulating or evaluating densities of various kinds. And then once that model is specified and data are given, then it allows you to, those algorithms become available.
So do you have any references for this that people can follow?
Yeah, there's a whole list of references at the back of the talk. I'll make sure that you have a copy of the slides. I might be able to just, can I cut and paste this whole thing into the?
Maybe in the chatting room, yeah.
I'm gonna just cut it. I'm just gonna paste it into the chat. How's that? That's crazy. Check that out.
Let's see.
Wow, there's a whole list of.
Oh my God, a lot. I see.
That's a lot, but I mean, that's just the whole set. But
if we go your GitHub, maybe can we?
Yeah,
I'll
send the.
Yeah.
And these references and others are available there as well.
Oh, I see.
So I have a small question.
Yes.
So you already, and first of all, I'm very appreciative for your very good talk.
Thank you.
And my question is that you already mentioned about advantages of using PMCMC, but I would like to know much about the advantages of PMCMC, like in practical sense. When you simulate the model using data, what's the main merit of particle filtering or PMCMC?
So the main merit, so particle MCMC is basically Metropolis Hastings, right? But where the likelihood evaluation is done using a particle filter. So the main advantage is that the particle filter is a efficient factorization of the likelihood, right? And so it reduces that integral that I showed you, which is a very high dimensional integral, into a series of much lower dimensional integrals. And it, there are other methods for evaluating those integrals, but in general, when you have a nonlinear, non-Gaussian process, perhaps with environmental forcing or whatever as well, the flexibility of the particle filter is very useful, right? Because it doesn't make any assumptions about the nature of that. So it can be used to integrate and compute the likelihood, and then you can use that in a Metropolis Hastings step. So the issue, of course, in all, I mean, it is an MCMC approach, right? So the issue is getting it to mix and getting it to converge. Typically, the mixing is the biggest problem. So the likelihood surfaces associated with these kinds of dynamic problems, especially if the data series gets long, is that the likelihood surfaces become complicated, right? They can have ridges that are nonlinear, that are curved. They can have multiple peaks. They can have very narrow valleys and so on.
Mm-hmm.
And so particle MC is not particularly well designed. I mean, you need to have a well-designed proposal distribution in order to be able to sample those things effectively, right? Mm-hmm. And it can be tricky. For that reason, I usually suggest that people first try to identify the high regions in the likelihood surface using a likelihood maximization technique, like iterated filtering, just so that they get themselves up onto the likelihood heights before they begin many different chains PMCMC. So running multiple independent chains is also a good idea. In many of these, there are techniques that exist now, Hamiltonian Monte Carlo, for example, that are extremely powerful for dealing with these complicated likelihood surfaces. But in these kinds of problems, we often run into difficulties with those methods because they require continuous variables.
Right.
And in many situations, the discreteness of the individuals is an important feature of the dynamics. And so that makes things difficult there. I will say too that the particle MCMC can be expensive in the sense that you may need many particles in a particle filter to get a likelihood calculation which is accurate enough. Well, it's not the accuracy, it's the precision, precise enough to be useful for MCMC, right? Because the Metropolis Hastings acceptance rejection, right? When that's noisy, right? Because the MCMC particle filter, is a Monte Carlo algorithm itself, that will be noisy. And so unless you get the Monte Carlo variability quite small, you can get stuck.
Right.
Right. You understand. So anyway, so those are some thoughts about PMCMC, just some observations. I'm happy to talk more with you about it if you're interested. There's lots that can be said, but I just try to throw out some general ideas or some general observations.
Thank
Hope
you.
that's helpful. Sure.
Okay. Yeah, please. Yeah, if you have a question, Hyuk, go ahead.
Okay. I have a long question. Thanks for the great talk, Professor Kim. And if I understood correctly, you say simulation-based likelihood computation by plug and play method, right? And then actually, I'm using such approach for inferring some parameters associated with hidden process with partially observed data. And in terms of simulating for estimating parameter in MCMC method, we have to simulate all the time at each iteration for MCMC. So it takes too much computation cost in common. So
you mean the entire time series you have to simulate?
Yes.
I see.
Using Gillespie algorithm, let's say.
Oh, dear.
So is there any other way to reduce computational cost when we use simulation-based likelihood?